Hi friends, welcome to your lesson on vision processing and visual interpretation. Your notes say that this is 3.1 and 3.2 possibly, but this is actually 3.3 and 3.4. So we're going to start off with talking about what light energy is visible to humans before we get into how the eye works and processes the light. So the part of the spectrum that's visible to humans is actually really small. So we can see light waves with a frequency of a little less than 400 nanometers and a little more than 700 nanometers. So if you look at the left-hand side, the visible spectrum, you can see it goes from gamma rays, X-rays. This is the electromagnetic spectrum, the whole thing along the bottom from left to right. But the visible spectrum is in between UV rays and infrared rays. So that very, very small part, that's what's actually visible to humans. And we see it as Roy G. Biv, the colors of the rainbow. So what information do light waves give us? Well, light waves give us wavelength and amplitude. Wavelength tells us what hue or color you're seeing, and amplitude tells you how bright the color is that you're seeing. So wavelength is how far apart the tops or the crests of the wave are. The amplitude is how tall the waves are. So if you're taking like a physics class, you're going to get into wavelength and amplitude in very detail, and you're going to do lots of math with it. But for this course, we're just talking about how short or long they are. Long wavelength means the crests are really far apart, and you're going to see reddish colors, and short wavelength, they're close together. You'll see bluish colors. That is also something that you learn in earth science when you're learning about how light travels from bodies in the solar system. So light traveling with a shorter wavelength is going to show up bluish, and longer will be reddish. And then big amplitude, all from the bottom to the top of the crest, is going to have be bright colored, and a small amplitude is going to be dull colored. So the next part is this whole table that takes up pretty much the whole front of your notes on what structures of the eye help focus the energy. First, we're going to start off with the cornea. The cornea is the eye's clear protective outer layer covering the pupil and the iris. Light enters the eye first through the cornea. The pupil is the small adjustable opening in the center of the eye through which light passes. The iris is a ring of muscle tissue that forms the colored portion of the eye around the pupil and controls the size of the pupil opening by expanding and contracting over the pupil. The lens is the transparent structure behind the pupil that changes shape to help focus images on the retina. And the lens changes shape by having it has a curvature and a thickness in a process called accommodation. If the lens focuses the image on a point in front of the retina, you see near objects clearly, but not distant objects. This is called nearsightedness or myopia, and can be remedied with glasses, contact lenses, or surgery. Hyperopia is farsightedness, and that's a result of the lens focusing light past the retina. The retina is the light-sensitive inner surface of the eye, containing the receptor rods and cones, which we'll talk about in a minute, plus layers of neurons that begin the processing of visual information. So if you see in the diagram out to the back, you see the retina up at the top on the right, and then at the bottom on the right, you see the optic nerve to the brain's visual cortex. The retina is located along the back of the eye and contains the sense receptor cells. These are rods and cones that receive the incoming light waves. Light waves are transduced into neural impulses by the rods and the cones, and then they're passed on to bipolar cells and ganglion cells. Really, you just need to know that they're passed on to cells. And the rods are located along the retina's outer periphery. So rods, outside, cones, center. And cone starts with a C, center starts with a C. Cones cluster in and around the fovea, which we'll get to in a second. That's the next thing that we'll go over. And in dim light, cones become unresponsive and we are unable to see color. Many cones have their own hotline to the brain. One cone transmits its message to a single bipolar cell, which relays the message to the visual cortex. That's where a large area receives input from the fovea. So the cones do have their own hotline to the brain, where the rods do not. The fovea is the central focal point, fovea focal, in the retina around which the eye's cones cluster. This is the area of greatest visual acuity or sharpness of focus. The optic nerve is comprised of the axons of the ganglion cells, and it leaves through the back of the eye, as you can see in the diagram on the bottom right, and it carries the neural impulses from the eye to the brain. And you know this is happening super duper fast for our brains to be able to register what we're seeing. The blind spot is the optic disc. The optic disc is the point at which the optic nerve leaves the eye, creating a blind spot. Because no receptor cells, meaning rods or cones, are located there. What happens to the neural impulse after it exits the eye? The optic nerve carries the impulse to the thalamus and onto the visual cortex of the occipital lobes. So the optic nerve in the back at the bottom carries that impulse to the thalamus and to the visual cortex on the occipital lobe. The Young-Helmholtz trichromatic, which means three colors, tri meaning three, chromatic meaning color, theory, is the theory that the retina contains three different types of color receptors, or cones. Color receptor is a cone. One most sensitive to red, one most sensitive to green, and one most sensitive to blue, which, when stimulated in combination, red, blue, and green, can produce the perception of any color. So, what about people who can't see color? This is a cool diagram. The photo on the left shows how people with red-green deficiency perceived a 2015 Buffalo Bills versus New York Jets football game. Go Bills! Everyone looks like they're on the same team on the left-hand side, according to a colorblind fan. On the right-hand side, you can see how the game looked for those with normal color vision. You can see the red and the blue. The red and the green versus the Jets are green and the red is the Bills. So some characteristics of colorblindness: about 1 in 50 people are colorblind. Males are more affected since the deficit is genetically sex-linked. I used to have a roommate who was colorblind, and I used to mess with him all the time and be like, what color is this? What color is this? What color is this? And we would do funny things with his clothing. He caught on pretty fast, but he just couldn't see green. Most people are not actually blind to all colors. They simply lack functioning red or green sensitive cones, or sometimes both. So my friend could see red but not green. Vision in a colorblind person is monochromatic, one color, or dichromatic, two color, and seems normal to them. So they just don't see red or green, and it just seems normal because they don't know what red or green are. Exam tip: there's typically a question about color blindness on the AP exam, and it's important to remember that it's most prevalent in males and that the red-green cone deficiency is the most common form. So think about the Jets and the Bills. The Jets are green, the Bills are red. Okay, so we're moving on to the bottom of the first page. To the top of the back page. And we're getting into the second part of the lesson in just a minute when we get to the table on visual perception. So this will wrap up part 1. What is the Herring opponent process theory? So this is a theory that cone photoreceptors are paired together, red-green, blue-yellow, white-black, and that enables color vision. The activation of one color of the pair inhibits the activation of the other. So for example, some cells are stimulated by green and inhibited by red, and others are stimulated by red and inhibited by green. And that's the fact that some are inhibited is what allows you to be able to see that other color. So let's try it. And I think it's going to work in the video. And if it doesn't, we'll do something in class with this. So stare at the center of the flag for a full minute. You're going to need to pause this because I'm not going to sit here and wait quietly for a minute. But pause it for a full minute and then go to the next slide. The opponent process theory continued. So now stare at the black dot. What do you see? You've just looked at the color one for a minute and you're going to look at this for a couple, 10 seconds. And what do you see? Did you see a red, white, and blue British Union Jack, which was the symbol you looked at? Here's the answer for why the green became red. First, you stared at the green bars, which tired your green response. Then you stared at the white area. White contains all colors, including red. And because you had tired your green response, only the red part of the green-red pairing fired normally. The same happened with the blue-yellow pair and the black-white pair. Super duper cool. So how does color processing occur? The retina's red, green, and blue cones respond in varying degrees to different color stimuli, which is what the Young-Helmholtz trichromatic theory suggests. The cone responses are then processed by the opponent process cells as Hering's opponent process theory proposed. So really, let's break that down because that's very wordy. The retina's red, green, and blue cones respond in varying degrees to different color stimuli. Okay, so red, green, and blue cones responding, and then those cone responses are processed by cells. Last part, what is parallel processing? It's a way of thinking about many aspects of a problem simultaneously. This is the brain's natural mode of information processing for many functions, including vision. And it works by the brain, it delegates the work of processing motion, form, depth, and color to different areas. So after taking a scene apart, the brain integrates these different subdimensions into one perceived image, which is so cool. So when we look at something, we're looking at the motion, form, depth, and color, and then processing it to make it one thing. But it happens so fast, we don't even realize that that's happened. So how we recognize faces using parallel processing. This is really cool. This is not on your notes. It's just a neat little blurb. So to recognize a face, your brain integrates information projected by your retinas to several different visual cortex areas and then compares it with stored information, which enables your fusiform face area to recognize the face as grandmother. And some supercells, actually nicknamed grandmother cells, do appear to respond very selectively to one or two faces in 100, which is really cool. So in the end, how do we see? As you read these words, the letters reflect light rays onto your retina. That triggers a process that sends formless nerve impulses to several areas of your brain, which integrate the information and decode its meaning. The amazing result, we've transferred information across time and space from this page to your mind, and that all happens instantly, effortlessly, and continuously is so awesome. Part 2, visual perception. So we're starting off visual perception with Gestalt. So we learned just in the first part all about how the eye works. Now we're going to learn about how it perceives things. This is so cool. So Gestalt, we're bringing him back from Unit 1. Early in the 20th century, a group of German psychologists noticed that people who were given a cluster of sensations tend to organize them into a Gestalt, which is a German word meaning a form or a whole. Gestalt psychologists believe that in perception, the whole may exceed the sum of its parts, and we'll get into what that means in the next couple slides. So the Necker cube illustrates a Gestalt. If you look at this diagram, you can see the blue dots, the blue circles, and then you can see a white cube. The individual elements of this figure, called a Necker cube, are really nothing but 8 blue circles, each containing 3 converging white lines. When we view these elements all together, however, we see a cube that sometimes reverses direction. It depends on how you look at it, whether you see the cube popping out or pushing in. It's really, really cool. The Necker cube is an excellent vehicle for understanding the distinction between sensation and perception, because only the visual stimuli are the blue wedges. That's sensation. The circles, lines, and cube are all products of your mind and are not on the page. That is perception. Okay, more Gestalt. What is figure ground? The organization of the visual field into objects, the figures, that stand out from their surroundings, which is the ground. Can you detect figure and ground? See if you can reverse the figure and make it the ground. Now try making the ground the figure. Your choice of what is figure is what creates this illusion. So if you look at figure as being the black, you're going to see something different as if you look at figure being the white. It's really cool when you can adjust your eyes to see the two different things. So Gestaltists apply rules for grouping to perception, and they do that when we apply. These are rules that we apply even as infants and even in our touch perceptions. They illustrate how the perceived whole differs from the sum of its parts. So if we looked at the slide before with the two black parts, which you might have seen faces, or the hourglass thing, that's what I'm talking about, the whole in its parts. Proximity is a Gestalt law of grouping that states we group nearby figures together. So when we look at this, we do not see 6 separate lines, but we see 3 sets of 2 lines. Continuity is a Gestalt law of grouping that states we perceive smooth, continuous patterns rather than discontinuous ones. So this is really cool. This pattern could be a series of alternating semicircles, but we don't perceive it that way. We see it as 2 continuous lines. One is a wavy line and one is a straight line. We don't see it as half circles that are alternating. Closure is a Gestalt law of grouping that states when we see something, we will fill in the gaps to create a complete whole object. Thus, we assume that the circles on the left are complete, but partially blocked by the illusory triangle. Add nothing more than little line segments to close off the circles, and your brain stops constructing a triangle. It's so cool. We see the triangle instead of the 3 circles with little bites taken out of them. So let's look at this. This is cool. How can this be happening? How are the principles of closure and continuity illustrated in this image? Well, this is called the impossible doghouse. From the photo angle in the try it picture, which was the last slide, the Gestalt grouping principle of closure leads us to perceive the boards as continuous. So last, depth. This isn't last, this is moving on though. What is depth perception and how have we tested for it? So depth perception is the ability to see objects in 3 dimensions, although the images that strike the retina are 2-dimensional, and this is what allows us to judge distance. There were Eleanor Gibson and Richard Walk in 1960 designed a series of experiments in their Cornell University lab using a visual cliff, a model of a cliff with a drop-off area that was actually covered by a sturdy glass. And the visual cliff demonstrated that 6 to 14-month-old infants, when they were placed on the edge of the cliff and coaxed by their moms to crawl out onto the glass, most infants refused to do so. Even like coming to their mom, they refused, which indicated that they could perceive depth. Okay, what are binocular cues and how do they help us judge depth? Binocular cues are depth cues, such as retinal disparity and convergence, that depend on the use of 2 eyes. As an object becomes closer or farther, both binocular depth cues operate to help us judge distance. So let's try it. Hold your 2 index fingers about 5 inches in front of your eyes with their tips half an inch apart. Now look beyond them and note the weird result. How does retinal disparity work? by comparing retinal images from the two eyes, the brain computes distance. The greater the disparity, the difference between the two images, the closer the object. Convergence is the inward angle of the eyes focusing on a near object, which when you held your fingers out from your nose, you probably saw 3 fingertips and your eyes went inward. That's because of convergence. All right, so hold your textbook or other written material out at arm's length and focus on the words on the page. While maintaining your focus, you may have to blink. Slowly bring the book or page closer and closer to your eyes. Do you feel slight pain or tightening around your eyes? As you probably did, as the 4 muscles surrounding the eye work to move the eye to focus, they send signals to the brain that the object in front of you is getting closer. Cool stuff. Monocular cues, and how do they help us judge depth? So how do we judge whether a person is 10 or 100 meters away? Retinal disparity won't help us here because there won't be much difference between the images cast on our right and left retinas. At such differences, we depend on monocular cues, depth cues available to each eye separately. So binocular means 2, bi meaning 2, and monocular, mono meaning 1. All right, so we're getting into moving down some cool stuff. Relative height, this is all about perception and how we perceive things. We perceive objects higher in our field of vision as farther away. Because we assume the lower part of a figure ground illustration is closer, we perceive it as the figure. Invert this illustration and the black will become ground like a night sky. And we'll perceive it as farther. Relative size. If we assume 2 objects are similar in size, most people perceive the one that casts the smaller retinal image as farther away. Again, if we assume 2 objects are similar in size, most people will perceive the one that casts a smaller image, the one that looks smaller, as being farther away. So this is funny, this cartoon. I have no depth perception. Is there a cop standing in the corner? Or do you have a tiny person in your hair? Interposition is when one object partially blocks our view of another, we perceive it as being closer. The deer blocked the tree trunk, so the tree trunk seems farther. Relative motion, as we move, objects that are actually stable may appear to move. If while riding on a bus, you fix your gaze on some point, say a house, the objects beyond the fixation point will appear to move with you. Objects in front of the point will appear to move backward. The farther an object is from the fixation point, the faster it will seem to move. You probably have experienced this when you're sitting at a red light and all of a sudden it feels like your car is moving, but really it's the car next to you moving. It's because you're not focused on a fixation point, so it seems like everything's moving. It's such a freaky feeling too if you're driving. Linear perspective. So parallel lines appear to meet in the distance. The sharper the angle of convergence, which is where they come together, the greater the perceived distance. So the picture on the left compared to the picture on the right has a greater angle of convergence, so looks farther than the left. So the yellow sign and the car, which are farther away from where the 2 parallel lines of the road seem to come together, are perceived as close. The car in this image is closer to where the 2 parallel lines seem to come together and is perceived as farther away. All right, so light and shadow. Shading produces a sense of depth consistent with our assumption that light comes from above. In the inverted view, the hollow will become a hill, which is really cool because of where the light's shining on it. Oh my goodness, tongue twister. What is stroboscopic movement and the phi phenomenon? Our brain perceives a rapid series of slightly varying images as continuous movement. That is called a phenomenon called stroboscopic movement. We construct we construct that motion in our heads just as we construct movement in blinking marquees and holiday lights. So we perceive two adjacent stationary lights blinking on and off in quick succession as one single light moving back and forth. Lighted signs exploit this phenomenon with a succession of lights that creates the impression of, say, a moving arrow. Perceptual constancy is a top-down process that recognizes objects without being deceived by changes in their color, brightness, shape, or size. Regardless of the viewing angle, distance, and illumination, we can quickly identify people and objects quite quickly. Even if the image on our retina seems changing, our brain can keep it constant. So this is really cool, that perceptual constancy, recognize objects without being deceived by changes in their color, brightness, shape, or size. Color constancy is when we perceive familiar objects as having consistent color, even if changing illumination alters the wavelengths reflected by the object. Brightness constancy is when we perceive an object as having a constant brightness, even as its illumination varies. This perception of constancy depends on relative luminance, which is the amount of light an object reflects relative to its surroundings. Shape constancy is when we perceive an object as having an unchanging shape, even while our distance from it varies. And size constancy is when we perceive an object as having an unchanging size, even while our distance from it varies. So all these constancies, we perceive it as unchanging even when the distance varies. So we assume a car is large enough to carry people even when we see its tiny image from 2 blocks away. So to our eyes, it appears an object is moving away from us, is getting smaller. But our brain understands that friends and cars don't shrink and interprets the visual input as an increase in distance, which is a perception. The last part is the Ames room summary. So how does the Ames room alter our perceptions? The distorted room designed by Adelbert Ames appears to have a normal rectangular shape when viewed through a peephole with one eye. The girl in the right corner appears very large because we judge her size based on the false assumption that she is the same distance away as the girl in the left corner. Well, this works because, as the diagram reveals, the girls are actually about the same size, but the room is distorted. So you see the back wall is moved up at an angle, which makes the girl on the right look much bigger. Perceptual adaptation is the ability to adjust to changed sensory input, including an artificially displaced or even inverted visual field. I know this was a long lesson, 2-part visual. You're going to have lots of practice and some cool work with MC Escher's drawings on it. I can't wait for you to get to it. If you feel like taking a peek ahead, feel free. There are links on the website. Oh, whoops, one more slide. Can we adapt to inverted vision? Oops, missed, thought researcher Hubert Dolezal as he attempted a handshake while viewing the world through inverting goggles. But believe it or not, kittens, monkeys, and humans can adapt to an inverted world, which is really cool.